All right, folks, let's break into chapter 4 here and talk about metabolism or energy. So chapter 4 will be mostly kind of about cellular respiration, and then chapter 5 is about photosynthesis. So we'll split those up a little bit. So if we just kind of jump right in, when we're talking about energy in cells, we're talking about metabolism. We're probably going to be talking about ATP a lot, and so remember ATP is adenosine triphosphate. The way it works, and again, I just remember that I forgot my pad for writing, so you'll just have to bear with me as I use a mouse to write stuff. Let's see, let me clear this out. So you've got adenosine triphosphate, and so what that looks like is an adenine. Maybe a better way to write this would be just put the A in 3 P's. The adenine with 3 phosphate molecules attached. And where the energy is in these bonds. So in order to create these bonds, you have to put a lot of energy in there to stick those 2 phosphates together. The one we are most concerned with is the energy in this phosphate bond. What's going to happen during metabolism, first we're going to create some ATP, but then what we're going to do is we're going to break that ATP down into ADP, which is an adenine. I'll put the T in there to be consistent, with 2 phosphate molecules. So adenosine diphosphate, and then we'll have that other phosphate molecule out in the cell. Maybe it's in the cytoplasm or outside the cell or wherever. And so when we break that bond and release that phosphate, that's where we get our energy in order to do work, right? And so let's see, let's jump back here and start talking about some of those processes. So at least here on Earth, most of the energy that comes in is going to be from the sun. We used to say all energy comes from the sun, but now we know that there are organisms that live at the bottoms of the oceans around these thermal vents that have never seen sunlight, and it's so deep that the sun doesn't really have an effect down there. And so most of the energy comes from the sun, not all of it. And so that sun is usually picked up by producers, primary producers, which are plants, and then consumers eat those plants. Producers will sometimes die and decompose. Consumers will also decompose, where fungi, bacteria, and worms will consume those things and then release that energy back out. Not always in the form of heat. Sometimes they'll release it back out as CO2 or whatever, and then you just continue this cycle over and over and over. Right, and that's the bioenergetics. The pathways that we'll focus on. So this first one, right, so CO2 plus H2O gives you sugar, C6H12O6, and oxygen. That's photosynthesis, right? So plants take in CO2 and water, they make sugars, and they release some oxygen. This one down here, right, so if I take in sugar and some oxygen and I release some water and some carbon dioxide, that's what you do, right? That's respiration. And so if you notice those two equations, right, they are the same, just essentially flipped. And so that's why we couple these 2 chapters together. Right? So photosynthesis, essentially, if you flip it around, it's respiration, and if you flip that around, it's photosynthesis. So something to kind of keep in mind. So just memorize this formula and then switch the arrow, and you've got the other. Right? When we go through chemical reactions, they can either be anabolic or catabolic. Anabolic reactions are reactions that tend to put things together. I usually use the kind of anabolic steroid. Most people know what steroids are a whole class of molecules. Some steroids, though, are the ones like people might use in the gym to build muscle. So anabolic pathways build things up. Catabolic pathways break things down. So if I have, for instance, a bunch of amino acids and I want to put them together to create a protein, that's an anabolic process. If I eat a steak and I take that protein that is the steak and I break it back down into its amino acids, then that's a catabolic process. Right? All of these things will tend to follow the laws of thermodynamics, right? Which essentially tells us things like we can't create energy. We can only change its form. So when the energy from the sun comes in, we can change that energy from light and heat energy into chemical energy, or maybe eventually into motion, but the energy wasn't created, right? It reached us from somewhere, we transformed it around into something else, and then that was that, right? So notice here we've got chemical energy that we can turn into kinetic energy. So you eat ice cream, that gives you energy to do work. Or we've got light energy that we can turn into chemical energy in the form of sugars in plants. So we just change the form of the energy. We don't create it and we don't destroy it, right? Whatever is there is there, right? Usually if it's lost, if it's not used to do work, it's lost as heat, right? That's how we maintain our body temperature, for instance, 98.6. Is because we consume food, we don't use all of the energy in that food to move muscles and things, and so some of that energy is lost in the form of heat, and that helps us maintain body temperature. Okay. They talk about a light bulb here. Some of the most inefficient light bulbs are the ones that get really hot when you touch them, right? So they're not turning all of the energy that reaches them into light energy. They're turning it into heat as well. And so that's a sign of inefficiency. If you know anything about cars, you'll know that a diesel car is more efficient than a gasoline car. And you would also know that because diesel engines are more efficient, they run a little cooler. If you ever had a diesel engine in the wintertime, it takes forever for the heat to get hot because of that reason, right? They're just more efficient. Potential and kinetic energy. Potential energy is stored energy. Kinetic energy is the energy of motion. So almost all books use this example of water behind a dam. So as the water is backed up behind the dam, it's got the potential to do work. It's not doing anything, though. It's just kind of sitting there. And then as it flows through the dam and turns the turbine, it's kinetic energy at that point so that it can eventually make something like electricity. Sometimes books will show like a bowstring. So when I pull a bow back, I've got a lot of potential energy in that bowstring. When I release it and it shoots the arrow, that's the kinetic energy, right? So potential and kinetic energies. Processes that are exergonic or endergonic. So usually exergonic pathways are those that are spontaneous. So they just tend to happen. They're usually high energy molecules going to low energy molecules, and they tend to release heat when they do it. Endergonic reactions are the opposite. So low energy molecules that need to be turned into high energy molecules, and they tend to absorb heat or energy. Maybe an example of an endergonic reaction is if I have amino acids in me and I'm building up proteins, right? That's an endergonic process, right? It needs energy to occur. The exergonic processes might be turning sugar into, so taking glucose and turning it into energy, right? Exergonic. And just because they're spontaneous doesn't mean that they always are spontaneous. Sometimes enzymes will cause them to happen more quickly. Sometimes I think students confuse the word spontaneous with fast, and that's not true. If I leave sugar on my bar at home, eventually it'll break down and become something else, like it'll release some CO2 and some water, and like if you just left it there long enough. It might take years, but it would happen, and that's spontaneous. But when we consume sugar, we have enzymes in us that help break it down, and so you don't have a year to break down the sugar that was in the coffee you drank this morning. It needs to happen fast, and so you have enzymes that can help that to occur. One of the ways they do that is by reducing this activation energy, right? So if this purple line is the amount of energy it takes for a reaction to occur without an enzyme present, then the red line is the energy that it would take to kick off a reaction without an enzyme. And so notice here they're showing you a reaction that has a low energy in the molecules at first, and then it has higher energy at the end. So again, you can create those same products with less energy input by the use of enzymes. Right? Enzymes are super specific. They work kind of like lock and keys, and they're named usually for how they work or what they work on. For instance, if maybe you are lactose intolerant, that's because there's an enzyme that's specific to the lactose sugar called lactase, and maybe you don't have that, right? Maybe you don't produce that. And because lactose and lactase lock and key, right, that's how they work, no other enzyme in you is going to function to break down the lactose like you should. Right? So very, very specific enzymes tend to be. Let me pause this. My phone keeps on ringing. All right, I'm back. It's one of the reasons I don't like doing recordings in my office, because I always get phone calls. Right? I would rather do them at home. All right, so this is just showing you that kind of lock and key specificity of enzymes, and they really do need to work that way, right? So although this is not always true, typically one key unlocks a door, right? And if any other key is introduced to that lock, it doesn't work. Same thing with enzymes and substrates. So one enzyme breaks down a substrate or builds it up, right? So typically when we think about enzymes, we're thinking about breaking things down, but enzymes also help build things up. And you are a very enzymatically driven organism. You have a bunch of enzymes in you. The benefit of enzymes, though, is that they're not broken down during the process. So if I have an enzyme that's responsible for building proteins, the enzyme is not degraded in the process of building proteins. So you don't need to produce a lot of enzymes. A very small amount will do. But if those enzymes become damaged, then you need to replace them. Right? We can control how enzymes work through things like competitive inhibition. So even though I just told you that enzymes are very specific, sometimes there are other molecules that will compete with them at the active site just because of chemistry, really. And so if something is competing with the substrate in an enzyme, then that enzyme is not going to function properly, or it's not going to break down the substrate. Sometimes that's purposeful as much as enzymes can be purposed. What I mean is, sometimes you don't want enzymes to function, right? Sometimes you don't want them to be active. And so in those cases, you could have competitive inhibitors that would stop an enzyme from working, right? You can also have non-competitive inhibitors. Those would be maybe inhibitor molecules that attach to a protein or attach to an enzyme somewhere else other than the active site, right? So if this, here they're talking about allosteric inhibitors. And so if you have an enzyme that has an active site, but you're not interacting with that active site, or you're interacting with some other part of that enzyme, when that inhibitor interacts with that site, it'll cause the enzyme to change, and then it can't break down that substrate like it normally would. So this enzyme is showing you allosteric inhibition, so inhibiting an enzyme from functioning. You also have allosteric activation. An example in us might be something like vitamins, right? One of the reasons that you need some of the vitamins that you need is so it interacts with that allosteric activation site and actually allows the enzyme to function, right? Without it, the enzyme won't function, right? And so those are just some of the ways that you can control how and when enzymes work. This also shows you some about feedback mechanisms. So again, another way to control how enzymes are working is, so notice here we have an enzyme with a substrate. That substrate gives you an intermediate, right, when it's broken down. And then when another enzyme breaks down that first the intermediate, you get the second intermediate. And then when that one is broken down, so you get these chain reactions of events, finally you get the end product. And so what can happen is if you get a buildup of too much of this end product, it can interact with the enzyme itself and prevent it from continuing to function. So maybe you build up too much. We could use sugars maybe as an example. So if you're to build up maybe not enough or too much of whatever, it'll interact with the enzyme and stop it from functioning. Right? These are typically called negative feedbacks because they stop something from occurring. A negative feedback is how your body temperature is controlled. Right? If you get too hot, you sweat, so you'll cool down. If you get too cold, you shiver, so you'll heat up. Right? Those are negative feedback loops. Right? And so this is just showing you how you can get feedback inhibition to prevent enzymes from just continuing to churn along. Right? Because if this enzyme just kept going and kept producing all these substrate and products, that might be wasteful. There might be no reason for that enzyme to keep doing that, right? You only need a small amount of whatever that substance might be, and so stop at a certain concentration. So then if we go into the first kind of process and look at glycolysis, if we break that word down, glycolysis, so really glucose lysis, glyco for glucose, which is a sugar, and lysis for to lyse, which means to break or split. So that's literally what we're doing in glycolysis. We're taking glucose, which is a 6-carbon molecule, and we're breaking it apart into 2 3-carbon molecules, and that's glycolysis. At the end of all of this, we're going to make some ATP, which I already kind of talked about. At the end of the whole process, we're going to make some ATP. Right? And so this is all that's happening in glycolysis. You've got this 6-carbon molecule. The first thing that's going to happen in glycolysis is you're going to break that 6-carbon molecule up into 2 3-carbon molecules. Right? And the way you do that is by investing a little bit of ATP. Right? And so all this time I've told you we're going to make some ATP, and then the very first thing I tell you right out of the gate is that we're going to invest some. Well, the way to think about that is, you kind of have to invest some money if you want to make some money. So here we have to invest some ATP in order to make all the ATP that we want. Right? And so, again, what happens is you take 2 ATPs, right? That energy allows us to break this 6-carbon molecule into 2 3-carbon molecules. Right? Notice that we have some phosphates here. Right? Those came from the 2 ATPs that we invested. And then we go through another series of reactions that you could spend weeks in biochemistry studying this stuff. But we're going to liberate some phosphates from this 3-carbon molecule, and we're going to collect some ATP, and we're going to collect some NADH. The easiest way really to think of ATP and NADH is just batteries. So a AA and a AAA, or a AA and a C, right? Whatever. They're just energy-carrying molecules that we're going to use to do work later. Right, and so if you notice, from this first process, which is glycolysis, we invested 2 ATP, and then we generated 1, 2, 3, 4. Right? So really we netted 2 ATP through this process. We invested 2, we got 4, right? And so we really netted 2 ATP, right? And so that's where some of the first ATP comes from in this pathway. Right? The other thing to remember about this is in organisms that have mitochondria, this is not happening in the mitochondria yet. It's happening outside the mitochondria in the cytoplasm. All right, so then we'll go on to the next kind of big cycle, which is called the citric acid cycle. And so what we're going to do is, notice this 3-carbon molecule, right? So if I go back to glycolysis, right, there's our pyruvates, which is that 3-carbon molecule generated in glycolysis. So when we start the citric acid cycle, we're starting with that end product that was before, right? So there's the pyruvate, the pyruvic acid. And so what we're going to do is use an enzyme to break off one of these carbons, right? And so there it goes, and it's going to form CO2. Right? You ever wonder why you exhale CO2? Right? There's why. Because you took the sugars in you removed a carbon from it, that carbon would combine with oxygen, and you exhale it out. Right? And so when that happens, that coenzyme A binds with that now 2-carbon molecule, and we just call it acetyl-CoA. Right? So we went from a 3-carbon to a 2-carbon molecule. right? Where'd the carbon go? We exhaled it, right? And so now that 2-carbon molecule enters the actual citric acid cycle. We're going to break off the other 2 carbons, right? So how many did we start with? We started with 3, right? We took off 1 there, and we took off 2 more here, right? And then in that process, we generate more ATP, right? And so really the big thing for me here for you guys is to kind of count these carbons and kind of know where you're at when that happens, right? So pyruvate, right? You break off a carbon, exhale it, and then acetyl-CoA goes into the citric acid cycle. You break off 2 more carbons, exhale them, and you generate some ATP. Right? So kind of, I know it sounds probably pretty detailed for you guys, but this is just kind of on the surface here, and I don't want to dive down too much in the details. Although there will be some of the quiz questions, but for the quiz questions, you can use your book and come back and answer some of those. Right? And so then what do we do with that, right? We have this ATP, this FADH2, this NADH, so we've got all these batteries, essentially, right? What are we going to do with it? Well, we're going to go through a process called oxidative phosphorylation. What in the world is that, right? To phosphorylate something just means to put phosphate molecules on it. That's it. We're just going to take phosphates and stick some phosphates on there. And so if you notice, when we go into the membrane, maybe of mitochondria, right? So mitochondria have 2 membranes, an outer and an inner. But notice here, NADH, FADH2, right? And so those are the molecules that we produced up here, right? NADH, FADH2. All right, so just connecting the processes. If I take that NADH, right, and contribute some energy into this protein that happens to be in the membrane of the mitochondria, then I can use that energy to pump these hydrogens across that membrane. And so what ends up happening is you end up with a bunch of hydrogens on one side of the membrane and very few on the other. Right? Another way to think about that is hydrogen is positively charged, right? Because typically hydrogen, most of the time it doesn't have the electron in the outer shell, and so because it's just a proton, right, it's positively charged. So if I end up with a lot of positives on one side of the membrane, that means I've got a lot of negatives on the other, right? And so these things want to trade places, right? So these positives want to come that way. Right, and so that's what they're showing you here, right? So you pump all these hydrogens on one side of the membrane, and then all of those hydrogens are going to come back across the membrane through this protein, and when they do, when those hydrogens come back across, that's going to give this protein the energy that it needs in order to make ATP out of ADP, right? And so again, just to kind of reconnect, where did the NADH and the FADH come from? Well, it came from the citric acid cycle. Right? Where did the pyruvic acid come from? Well, it came from glycolysis. Right? And so the other thing to notice is we need, where did it go? Right here. So we need oxygen, right, when we're going through cellular respiration. Well, why? And it's right there, right? And you can see this a little better in your book. But notice that you take those 2 hydrogens, and it's telling you half of an O2, which is an O. So if you combine, if you know what water is, it's H2O, right? So where did the oxygen come from in order to make the water, right? Well, it came from the oxygen that you inhale. Right? And that's really why you need to inhale oxygen, so that you can go through this cellular respiration process. Okay. All right, that's just something that takes some time. One thing I would encourage you to do maybe is to draw this out on your own. Even if you're looking at this picture, right? Draw it out on your own. You don't have to worry about the names of these individual proteins. But just generally understand kind of what's going on, and where some of these things came from, and that'll help you. Okay. All right. Something else that we can talk about is fermentation. So that's something that I do often. I make a decent amount of wine. I used to make wine even for one of the local vineyards here a couple years ago. So I was making wine for Shadowline Vineyards. And before that, I used to make my own and still do. It's something that I enjoy. But there's a couple different types of fermentation. So there's lactic acid fermentation. That's the kind of fermentation that occurs in your muscles. So you take that pyruvic acid and NADH and create that lactic acid and NAD. That's really the process. So if oxygen levels get low in your muscles, that's what will happen. And really the purpose there is just to keep regenerating that NAD so that you can get back and break down the pyruvate and continue to release energy, right? So here it's showing you that process, right? So glucose to pyruvate, right? That's the same, that's glycolysis, right? So nothing changes there. You don't need oxygen to take place for that. But in order to go from pyruvate to the lactic acid, you pump in some NADHs, right? You regenerate that NADH2 or that NAD, right? And by regenerating that NAD, that lets you break down more glucose. That's really the purpose of lactic acid fermentation, is to keep regenerating that NAD plus so you can break down glucose and make pyruvate. Right? That's the purpose. That's why we do it. The problem with it is it doesn't generate a whole lot of ATP. Right? So you're making just a couple ATP, whereas with oxidative phosphorylation, I think they talk about ATP yields here at the bottom. Right? So you're talking about, maybe 32 to 34 ATP, whereas with fermentation, you're getting just a couple, right? So not nearly as much. That's why you get tired, right? The other fermentation is alcohol fermentation, right? So in that, you're going to take the same pyruvic acid and you're going to break it down. You're going to release some CO2 and some ethanol, right? Here they're showing you the kind of in-between steps that acetaldehyde that gets broken down and makes ethanol. But essentially, pyruvic acid to CO2 releases some CO2 and makes some ethanol. Right? So when you're making wine and you see it bubbling on the top, all those bubbles are CO2. Beer, the reason it's carbonated usually is because of the CO2 that's produced during the fermentation process of the yeast. Right? And what else? So there are plenty of other pathways. So usually I talk mostly about the sugar pathways because they're the most efficient. And why does this thing keep getting blurry? I don't know what's going on with that. We'll just deal with it. But you eat all kinds of things. You don't eat just sugar, right? You eat proteins, you eat fats. And so there's plenty of ways for you to take those same molecules, proteins, lipids, and turn them into energy. It just kind of, where does it enter the system, right? So remember, we've got the glycolysis, pyruvate oxidation, citric acid cycle, oxidative phosphorylation. Those are the things I just went over. And so usually the carbohydrates will enter here in the glycolysis. Proteins can enter a little further down the chain. Fatty acids can enter a little further down the chain. And so that's why you can get energy from all of the different types of foods that you eat and not just from sugars alone. Right? And so that's kind of it in a nutshell. Certainly watch the other videos. Let's see, I think I've given you, so I've given you several videos that kind of break down these things in a little more detail. And hopefully that helps. And as always, if you have any questions, please